Grandma has this dress.
No, neither.
I feel like Harry Potter. Too librarian. Too Bono?
No.
No. Big no.
Yes.
I look a little bit like a bumblebee. How long you been in these parts?
If sadness had a color. I bought this? Your Honor, I object.
No. This is so much like my dad.
That is the ticket.
We like to think we're in control, that our minds are our own. But that's not true for this fruit fly. its brain has been hijacked by another organism, and it's not gonna end well. It all starts when the fly is innocently walking around, sipping on overripe fruit. It picks up an invisible fungus spore, which bores under its skin. For a few days, everything seems normal. But inside, the fungus is growing, feeding on the fly's fat and infiltrating its mind. At dusk on the 4th or 5th day, the fly gets a little erratic, wandering around. It climbs to a high place. Scientists call this behavior summiting. Then it starts twitching. The fungus is in control. The fly sticks out its mouth part and spits out a tiny drop of sticky liquid. That glues the fly down, sealing its fate. A few minutes later, its wings shoot up, and it dies. Now that the fungus has forced the fly into this death pose, wings out of the way, nothing can stop it. It emerges. Tiny spore launchers burst out of the fly's skin. Hundreds of spores shoot out at high speed, catching a breeze if the fly climbed high enough. They're the next generation of killer fungus. It continues for hours, spores flying out. These flies are in the wrong place at the wrong time. And if spores land on a wing, which they can't bore into, they shoot out a secondary spore to increase their chances of spreading. So, how does a fungus take control of a brain? At Harvard, Carolyn Elia is trying to understand that. She thinks the fungus secretes chemicals to manipulate the fly's neurons, maybe stimulating the ones that make flies climb. But don't worry, the fungus can't hurt humans. Scientists have tried to harness its power for our benefit, to kill flies in our kitchens and farms. They haven't had any luck, though. The deadly spores are actually pretty fragile and short-lived. It turns out, this lethal puppet master does only what it needs to for its own survival.
Support for KQED Live comes from Berkeley Rep and the San Francisco Symphony.
Disproportionate impact of
more than half of Black business owners
and disproportionate.
Somehow we always find a way to rise.
To the blueprint builders, to the backbones of every block, for the curators of the culture, and for generations to follow. You might fall, but never fail. Keep rising, keep rising, keep rising. Apply for business, marketing, and tech makeovers on us.
Good evening, everyone. My name is Esperanza Vaquiz, and I am the live events intern. For those of you joining us for the first time, welcome. KQED is NPR PBS member station. KQED Live is our multi-platform events program that produces screenings, talks, performances, and food tastings based on KQED's mission to inform, inspire, and involve. If you want to learn more, you definitely can at kqed.org/live. If we bring value to your everyday life, and I hope we do, I encourage you to consider becoming a KQED member. You can find out about how at kqed.org/donate. We want to thank the season sponsors that make KQED Live possible: SF MoMA, Berkeley Rep, Comcast Business, and San Francisco Symphony. Our next event is a Living's Tradition celebrating the Lunar New Year on February 1st. Come through to dive into the heart of local traditions with an interactive art installation, culinary delights, traditional attire showcases, and hands-on crafts. All right, now tonight's host is Laura Klivans, a science reporter with the Science and Climate Desk here at KQED, and the host of Deep Look, a Ultra HD wildlife and nature video series created by KQED and presented by PBS Digital Studios that explores big scientific mysteries by incredibly small. Please welcome Laura to the stage.
Hi. Thanks for my diploma. Okay. Hello, everybody, and welcome to KQED. We love to see the space so full and full of people who are excited about fungi. I know when I came into the lobby and I saw all that amazing variety on the table, I was like, this is the coolest entrance that I've ever seen when I've come in this building. I come in pretty regularly. Okay, so my name is Laura Klivans. As SB said, I'm a climate and science journalist here at KQED, and I'm also the host of Deep Look, which is our ultra high definition video show. You can find us on YouTube and other places. And what we do is we look at big science concepts by going very, very small. And we will be seeing some of the videos that we have created tonight. Tonight is super packed. We got an amazing range of people interested in mushrooms here. And we're gonna be talking about mycology, psychedelics, foraging, cooking mushrooms. We're gonna have a cooking demo. You're gonna get a little taste of some stuff. And most importantly, we're gonna be talking about identifying them properly. And then at the end of the evening, we are going to all get to go out and socialize with one another, like create our mycelium networks with each other, and the wonderful experts that we have tonight. And so maybe you even brought your own mushroom and you'd like to ID it, and we can do that later at the tables. Okay, so first up, we are gonna start with some Deep Look videos and three wonderful scientists, and they are Grace Stark, a naturalist and a PhD student at UC Berkeley, Damon Tai, a biotechnologist and naturalist. Yes, let's hear it for all these wonderful people. And, that's for a little quiet. And Gordon Walker, he is Gordon Walker, PhD. He's a biochemist, and he is fascinated by fungi on social media, which you might be familiar with. Welcome, everybody. All right, thanks for welcoming our crowd to the stage. I also want to highlight the incredible outfits everybody has on tonight. We've got, could you just show off the socks that you just pointed out backstage? We got fungi socks, pins, shirts, like just take note. There's some excellent fashion choices this evening. Okay, so to start off, and this is a question I want to ask everybody who comes on the stage, because it sounds like a pretty interesting story, but and think about it for yourself. But the question is, what is your mushroom origin story? Can we start with you, Grace?
For sure. I started off in an academic lab when I was 18. So I was extracting fungi from leaves, and they were just coming out on the plate as beautiful colors, and I was enamored immediately. So it's pure aesthetics, just gorgeous organisms. Specifically, there was this one that was fruiting on the plate. Xylaria, if y'all know, that just completely captured my attention. So I was pretty sold after that.
Thank you.
Yeah.
All right. Damon, how about you?
Mine's a little less aesthetically pleasing. I was out of food on a very long backpacking trip, say a 220-mile walk from Yosemite to Mount Whitney, and I hadn't planned my calories right because it was my first backpacking trip over 2 days. And I remember seeing fungi and being like, I wish I knew if I could eat that, because right now I'm basically just getting by on berries and the fish we can catch for 2 days. So I basically came home that year and was like, I got to figure this out. And so I started just trying to learn one mushroom a year, and then it just became an absolute obsession as I realized how much we didn't know about what was all around us, especially here in California.
Awesome. What about you, Gordon?
I was a kid in the woods of Quebec as a kid and saw this beautiful slimy green mushroom, which I know now is a parrot mushroom, but I was like dead convinced it was an alien. So I went to my grandfather and was like, it's an alien. He's like, no, it's Gliophorus pistaticus. And I was very disappointed that it was not an alien, but it kind of set me off in a lifetime fascination of fungi.
That's cool. It sounds like it might run in your family, because I feel like if I were to say to my relative, what is this, they wouldn't say that. Whatever you just...
Well, the other big part of it was when I was like 5, my mom and I found a puffball, and she knew enough to be like, we can take that home and eat it. So my first bite of mushroom, I was completely sold on, like, all right, I got to find out more about these things.
Oh, cool.
It was 20 years off that I didn't think about them, and then it was brewing in college that actually brought me back into the fungal fold.
Nice. Okay, that's awesome. Okay, now also I want to get us all up to speed here, because I'm sure we have people who are super knowledgeable in the audience and some folks who are maybe like me, who are a little bit more of a novice here. So tell us, what really is a mushroom?
So in essence, a mushroom is the sort of reproductive fruiting body of the mycelium below. So mycelium is the sort of amorphous white hair-like threads that are in the soil. We have a very hard time telling where mycelium starts and ends, but we can look at a mushroom and it's very distinct. So that is the part that we kind of identify with, and ultimately its purpose is to disperse spores and help the mycelium to reproduce. Sort of at its core, that's what it is. It's like the fruit or a flower of a fungus.
Yeah, it's like analogous to the sexual fruiting structure that's going to create, a spore, or, like an apple would create a seed. It's the tissue that's creating the progeny.
Yeah, okay.
And for most of us interested in fungi, it's the one portion of their life that we can actually see. And as Gordon said, the majority of their life is spent as mycelium, or these small threads that are so small you can't see them with the naked eye. So this is really our only holding on point as kind of macro individuals that can see stuff. And that mycelium may last for a very long time, where that fruiting body is very ephemeral. So in this year, with all this rain, we're seeing mushrooms that maybe only fruit once every 5 to 10 years. So it's really a special event when you see a mushroom.
Great. Okay, thank you.
That's Xylaria on the left there.
Oh wait, that's the alien one you found when you were...
No, this is mine.
That was
this
her.
is my origin.
Your obsession.
Yeah, this one showed up in the plate when I cultured it.
Ooh.
It was really weird.
Very cool. Okay. Great. So what we're going to do is we will start off watching one of our first deep look video. Although some of you saw this one about the flies that sort of became zombies as you trickled in. But the next
Damon
one
used to have those in his apartment.
What?
Damon had those zombie flies in his apartment.
I brought one home from Point Reyes, and I had a fly problem, and for the next 7 years, I didn't have a fly problem.
Oh my God. Very interesting. You can unpack that afterwards with Damon.
I had a different problem.
So our first one that we're going to watch is about the cage fungus. So let's roll that video.
What is that terrible smell? Is it spoiled fruit? A rotten egg? A dead animal? It's actually coming from this cage fungus, which is in the absolute prime of its life. That horrible smell is part of an elaborate hoax to fake its own death. Like most mushrooms, what you see is only a small part of the organism. The vast majority is hidden below the surface in an expansive network of minuscule threads called the mycelium. Cage fungus is saprobic. The mycelium eats dead and rotting wood, digesting the cellulose and returning nutrients to the soil. When the weather is warm and wet and there's plenty of wood around, the mycelium creates an egg, this brain-looking thing that pops up above ground. A white leathery membrane protects the egg as it grows and stretches until it cracks open. The spongy lattice bursts outward. After a few hours, it looks and smells like a basket made of decaying flesh. The interior surfaces of the lattice are coated in a sickly brown slime called gleba. That horrendous odor emanates from those sticky globs. The open lattice shape helps the stench spread in the breeze. But to a fly, that reek is irresistible. It screams rotting carcass, a delectable meal, or a place to lay their eggs. The gaps in the lattice are just the right size for the flies to pop in and out. They traipse around, gobbling up the goo. The flies love it, but what's in it for the fungus? Well, the fungus needs something that the flies have: wings. While many mushrooms spread their spores by releasing them into the wind, the cage fungus gets the flies to do all the work. As they feed, the flies unintentionally down millions of the fungus's microscopic spores, hidden inside the dark slime. The gleba also clings to the flies' feet. When the flies lift off, they take the spores with them, spreading them far and wide. After about a day, the lattice starts to collapse. Having successfully spread its spores, this is the end of its story. Or is it? When the conditions are right, the fungus will pop up more eggs year after year, continuing its deception for the sake of future generations.
Nice. Now, Damon, you really helped us with that video. Where can we find cage fungus around here?
So cage fungus in the Bay Area is in just about every man-made natural landscape you'll look at. So think Golden Gate Park, Lake Merritt, and especially those portions of the parks that have wood chips. So this is a wood decaying fungi. And so any place you've seen wood chips spread about, start looking this time of year through about late spring, and you'll see them popping because they're hard to miss. Giant red flesh bag colored geodesic dome, easy to see.
You know, the first time I ever saw one, I actually thought it was trash. Like, my friend's child pointed it out to me, and I was like, that is trash. He was like, it's a mushroom.
Somebody dropped an organ.
Yeah. So you gotta be on the lookout for that shape. Okay, and then is it actually from here originally?
No, so this is like many of our urban landscape mushrooms. They're not from California, so that's why you don't see it if you go for a walk in our woodlands. So this is originally from the Mediterranean. So if you want to see this in its native habitat, you got to go to Europe.
Okay, and so one thing that strikes me when I consider the cage fungus is just how different it looks than a typical like mushroom, or what I think of as a typical mushroom, like what you are wearing on your lapel there. So can you all talk about that? Like, I think a lot of people think of a mushroom as that standard thing with the knob on top, but what are mushrooms? What can they look like?
So you have your sort of standard cap and stem mushroom, the classic toadstool like Amanita muscaria. There's a massive one out there that Damon found. The Cage fungus is an example of what's called a gastroid mushroom, but it's a thing where the spores are on the inside, so like a puffball, stinkhorns, bird nest fungi we're going to talk about next. There's jellies, there's corals, there's all sorts of little like carbon dots, and there's so many different styles of mushrooms out there. It's almost hard for us to decide like what constitutes a mushroom. It's a surprisingly contentious question for people to answer, and I think all of us can go back and forth.
There was some argument earlier.
Exactly. But I think this is a great time because you can see this whole presentation, then go out and look and sniff and smell and touch all the different types of mushrooms that we have out there.
Well, you just said something interesting, which is that you can touch them all, but I think people have heard some mushrooms can kill you. So what, is that true? Can you touch mushrooms? Tell me a little.
So yeah, you can touch any toxic mushrooms. The toxins don't pass through your skin. So there's a few things, and Damon and I have talked about, that might give you like a rash. But for the most part, you can touch any mushroom safely. End of sentence.
Okay. Yeah. And so just talking about mushroom biodiversity, can you talk about the amount of different types of mushrooms that exist?
I mean, it's hard to put a number on because it's been such an understudied field for such a long period of time. In fact, Grace and I are part of a crew this past year that have been doing collections all across California to sequence them and get a closer look. And what we're realizing is like, there's so much, and we don't have appropriate names on most of the ones out here. So when you talk about total species number of mushrooms, I don't know, Grace, do you have a good number for that?
I mean, there's thousands in California. That's the estimate for California. And that's like quite...
Yeah, what we know, the best
generous.
sort of guess worldwide, maybe there's like 40 to 50,000 species with probably maybe another 150,000 species to be discovered. That's a very hand-wavy number. Overall, there's probably about 5 to 10 million more species of fungi that are undiscovered. So mushrooms are just a small slice of the entire fungal pie.
Yeah.
A very small spice.
All right.
By the way, there's about 10 million yeast living on your head right now, if you didn't know that.
Which is part of the fungal kingdom?
Yes. Yes.
Okay, so we can find the fungus in the ground in wood chips, but also on our own bodies?
And inside of us.
And inside
of us.
And it's actually helping us for the most part, even though occasionally it can overgrow and cause a yeast infection. Same with dandruff. It's basically an overgrowth of that scalp yeast.
Wow. We have fungi to blame for that. Okay, cool. All right, thank you.
Or thank.
Or thank. Okay.
They can be our
great.
friends and our foes.
Nice. All right. So we're going to watch our next video. The next one is about the bird's nest fungi. All right, let's roll that one.
What can a single raindrop do? Send these mysterious eggs on a journey, for one thing. But these eggs have no yolk inside. Instead, they hold millions of spores that will spread this curious little mushroom called a bird's nest fungus. 2 of them would barely cover your thumbnail. They grow up on logs or twigs on the forest floor. Or mulch in your backyard. The spore sacs, known as peridioles, sit patiently in their splash cup, biding their time. When a raindrop hits the cup, the peridiole hurtles off in milliseconds and lands on the ground or a leaf. Bam! Pow! Zap! As it flies, this peridiole unfurls a cord and, with some luck, gloms onto a blade of grass. From the back, you can see how the cord wrapped around. A sticky bit at the end, called the hapteron, anchors it. The peridiole doesn't end up that far from home. It waits. dangling from this thin but surprisingly strong cord. It's made of thousands of entwined threads called hyphae, that same webby material a fungus grows underground. Yoo-hoo! Over here! A hungry deer nibbles the grass and takes the peridiole with it. As it wanders, it scatters the spores in its droppings and spreads the fungus to new frontiers. It's a slightly undignified journey, propelled by a plop and a drop.
Plop and a drop. Just a poetic way to leave that video. Okay, so what we saw there was a kind of interesting way of getting around for something that isn't really mobile. Can you talk more about that dispersal method and other ways that mushrooms spread?
Yeah, so, I mean, plop and a drop, right? So,
the official term.
Yeah, the official term.
Yeah.
So water dispersal is being used here to get the peridiole out into the world, and then herbivory, it's, evolved to be eaten by an animal that comes, and it will pass through the digestive system and, be excreted out in a different area. And that's very common for a lot of grassland fungi to attach to grass and be eaten. And herbivory is actually very common. Fungal strategy with the truffle-like fungi as well. They attract animals. But, fungi get around mostly, not mostly, but also by wind. Wind is actually most common in, the classic stem and cap mushrooms.
Like
and
the spores.
Yeah, the spores will just will be propelled out of the gill tissue, and so it's actually quite an active propelling process. A lot of fungi also will propel spores by osmotic pressure, like a cannonball. So they'll excrete spores out with a pressured force. So this is the main, purpose of creating the mushroom, is to get the spores dispersed. So, yeah, there are many strategies for dispersal.
Cool.
Yeah.
Great.
And these are definitely ones that you can see right now as well. In fact, if you're walking around and you find that cage fungus, it's a great reason to stop and look for other fungi. When you see one massive thing fruiting, there's usually other stuff. And the bird's nests love the same habitat as the caged fungus. So they're a really good way to kind of discover other diversity around them.
And when are the best times to find mushrooms? What are the conditions in which where we can find them?
Water.
When it's raining.
Yeah.
When it's wet.
But
What
you
is
can
it
find them
about them?
outside of that season too, depending on like what you're looking for and how you're looking for them. Like I've been out in the middle of summer with Damon, and I don't think I'm going to find anything. And he starts flipping logs, and there's a little bit of moisture on the bottom side of a log, and there's a tiny little micro mushroom there that turns out to be super fascinating. So it's really, they're around us all the time. It's just right now in winter, when there's a lot more moisture, that mycelium in the ground is swelling and producing big mushrooms that are able to push up and create big fruiting bodies that we can see.
Cool. I think what's interesting about the bird's nest fungi is that they're just so tiny, but they force me, once I learned about them, it forced me to really look down at the ground more and to observe and try to find them in my environment, because they can be found in backyards and all over the place, right? And so I'm curious, with your own journeys, as you've become, more and more enmeshed in this world of fungi, how do you see the world in a different way?
Geez, I can't walk down the block anymore. It takes like a half an hour. And that's the biggest thing. My wife can attest to that. But it also makes you just appreciate like the diversity of life that's around us so much more, even in the urban environment. A lot of people, when they think about going hunting for mushrooms, they go to the forest. They're like, I have to get someplace pristine. Like I spent 20 years around Lake Merritt, and I could go out every day and find a mushroom there. It just takes patience and like a desire to see, and that builds like some real joy into your life, because it's like an Easter egg hunt every time you find one, right? And it's a feedback loop. You're like, I just want to go do that again tomorrow. And pretty soon you just become Gordon, who's made a life of this.
Well, note the little clip-on macro lens that Damon has on his hat, because a lot of the stuff you're going to go find on an everyday basis may not be big enough to really move the needle for most people. You know, you're not going to find a giant, perfect porcini in your neighborhood necessarily. Maybe some places in the city, but for the most part, it's all about scaling down your expectations and increasing your focus on some of the little things when you're out of season.
Yeah, just enjoying the life that's all around you. It's really fascinating to see who you're inhabiting this space with, and who is, helping the ecology out around you.
And then once you start to see them too, I think one of the magical things about mushrooms is it forces you to see the rest of the ecology around them. Yeah. Because that mushroom is there because of that habitat, and that might be the plant that it's growing with. Maybe it's one of the chanterelles that's out there today, and it's growing specifically with coast live oak. Or maybe it's one of the bird's nest fungi, so that you've got to find it on dead wood already. It really helps you see all of these connections of all the kind of life forces around us. And I think that's one of the biggest joys that I've gotten over the years of starting to just be able to see fungi everywhere.
Yeah, like discovering microenvironments.
That's great. Cool. Thank you. Okay. So for our final deep look video of the evening, we're going to watch one that is about death cap mushrooms. Let's roll that.
As humans, we have an innate attraction to nature, a childlike curiosity. But beware of what you find in the shadows. In Northern California, something deadly is popping up in the forest. This poisonous mushroom is known as the death cap.
The forest was just damp and perfect.
Donna Davis went collecting mushrooms one day in 2014.
It's very magical when you're up there, and really is quite like a treasure hunt. Felt confident enough that I knew what I was doing.
But she didn't recognize the telltale cup-shaped tissue that grows on the bottom of the deadly mushroom. And she mistook young egg-shaped death caps for something she could eat.
I made mushroom soup. It was amazingly delicious.
One cap has enough toxin to kill a human being.
The next morning, I took a little nap.
For the first 6 to 12 hours, you feel okay. But meanwhile, the toxin is quietly destroying your liver cells.
The next thing I knew, I was in emergency care.
Death caps spread the same way other mushrooms do. Just like this harmless oyster mushroom, a death cap shoots hundreds of thousands of spores into the air. The spores drift into the shadow of California live oaks, where they grow filaments that attach to the tree's roots. Each fungus filament is about the width of a human hair. Under the microscope, you can see how the white fungus envelops the tree's pink root tips. This is called a mycorrhizal relationship. The fungus feeds on the tree's sugars and gives it nutrients in exchange. It's good for the fungus and for the tree. And that's how the death cap snuck into California from Europe on the roots of a decorative shrub in the 1930s. Researchers aren't sure why the death cap has evolved to be so lethal. What exactly is it protecting itself from? Humans, we're just collateral damage in some battle that we don't understand. Scientists like Anne Pringle from the University of Wisconsin are doing genetic testing on death caps to see how long they live. If death caps have a short lifespan, it might be enough to pluck them from the ground so the spores can't spread. Less opportunity for people to eat them.
I remember closing my eyes and seeing like this beautiful path. And all of a sudden, everything got really dark. And I thought, oh, I'm not supposed to go down this path.
Donna survived, but death caps killed 2 people in California that year.
It is really brutal. Nature has its own rules.
All right. Yeah. So very caught on video. Okay, so I think one thing that a lot of people know about mushrooms is that they can be lethal. And so how do you keep that in mind with the work that you do and the passion that you have for them?
So what I think about is out of, let's say, 50,000 hypothetical mushrooms, there's probably a couple hundred that are good edibles and probably a couple hundred that are really toxic. Here in California, the ones that present a real, clear and present danger to us as humans and as foragers are basically the deadly amanitas, which is the death cap and the destroying angel, Amanita ocreata. Amanita ocreata is native, endemic to California. It's not spreading, so you usually see it in the same spot every year, and it's just there. Whereas the death caps are invasive, and they're very competitive, so they're spreading around the state, and you're seeing them in more and more places. And so to me, that is the biggest danger. What you should do if you're out foraging and you're looking for mushrooms is just assume everything is not edible. Don't ever assume edibility. And even when you ask someone, don't say, is this edible? Just say, what is this? And that simple mind shift will help you stay safe because you're not assuming you can eat something. You're just trying to ask what something is. We live in a day and age where everyone has Google and a phone, and you can immediately take that name, look it up, and decide whether or not you want to take the risk to eat something like that yourself. So to me, that's sort of the important thing to keep in mind is don't assume that edibility and just be really careful because you want to start to be able to recognize what amanitas look like, and this is a great place to start. So if you can avoid amanitas, you've cut out 90% chance of getting poisoned by anything.
Right, yeah. And you're probably not going to eat Foliatina or tiny Gallerina that has, amatoxins are mostly in the larger amanitas, so.
Yeah, and the other thing that I always like to bring in, though, to discussion, because I know everyone's like, oh, death caps, scary, horrible, bad. As somebody that works in biotech, these things are amazingly useful. Without their toxins, we wouldn't understand a lot of cellular functions.
Typically RNA polymerase.
Yes.
The what?
RNA polymerase.
RNA polymerase. So the way that
all of the proteins in your body.
Yeah, the way the toxin works, it basically stops the central dogma of molecular biology from working. So the information is going from your DNA to RNA to the proteins that build up your cells. This toxin works on that pathway and it basically shuts it down. So being able to shut it down is really important as a scientist because it means you can then study it. And it's not just that toxin that death cap has. It has a couple other ones we use to pause out cells, the phallotoxins. It's got an amazing suite of toxins for a reason I still don't know.
And they're also exploring the use of those toxins as cancer treatments because you could selectively kill a cancer cell with some of these toxins. The thing that I think is important is a lot of people say, oh, if you see an animal eating a mushroom, it's safe to eat, right? No, do not assume that, right? You saw at the end there a slug chewing on something. Slugs and insects and even rodents have fundamentally different physiology than we do. The interesting thing about a death cap, though, is the toxins have evolved to kill animals, but it doesn't kill them until several days later. So it's not saving the mushroom. And it's still an open question is why did it evolve these really deadly toxins that do kill animals, but not in time enough to actually save a mushroom from getting eaten? We don't know.
There's a lot we don't know when it comes to mushrooms, even things that we really, really want to know like that.
There has been some recent work that was done on death caps that said most mushrooms need to find a mating partner, so you have to have two spores come together, and the death cap is somehow sort of able to double its own genome and use itself as a mating partner, and that's part of why it's spreading so quickly and it's so aggressive.
Wow, that's interesting. So what are some of the other uses that we have found, both for the environment, but also for people, that we can get from mushrooms, like what you were just talking about, even with a poisonous mushroom?
So one of the other ones I think that rises to the top a lot of times is anybody that's known somebody that's had a transplant of an organ. One of the primary compounds that's used for that, or medications, is cyclosporin, which comes from a fungus all of you are very well familiar with these days, which is the group of the cordyceps fungi, the fungi that attack insects. And so compounds...
The first video when people were trickling in, that one?
Exactly, well, similar to that.
Or it's also from, what was that show that everybody loves?
Last of Us.
The Last of Us, yeah. Yep. So remember, because fungi are inhabiting all of these different environments, so they've got this, and they've been doing it for a very long time, much longer than any of us, so they've got an amazing biochemical toolkit, and we're just starting to scratch the surface of it at this point in time. But it really comes down to understanding them and how they live, how they work, how they get away with not being eaten, how they eat others, that we need to figure out if we want to use their toolkit.
So in a very Last of Us type way, cyclosporin is an immune suppressor. So they give that to people who've had organ transplants to keep them from rejecting the immune thing. But in the ecology of that mushroom, it's using it to like invade an insect host and sort of trip up its like immune system so that it won't go crazy and kick out the fungus. So we just isolated that chemistry and are now using it for organ transplants.
And one thing to think about too is a lot of the mushrooms that we actually eat and consume have toxins in them that are keeping insects from eating them. But we destroy them through cooking and through other processes. And this is why cultures have long histories of the way they cook very specific mushrooms. They don't just cook them all the same, but it's usually appealing to ways to get rid of that toxin or that thing that might cause us harm, that is protecting the mushroom itself from being devoured by something else in the environment.
A death cap toxin cannot be destroyed.
Thank you, Grace.
Not heat libel.
Not heat libel, yes.
Okay. Any other safety PSAs that you'd like to share with this audience? Curious-minded.
Don't eat any mushroom you can't identify.
Okay.
Yes. Identify first.
Identify
Think about eating much later.
And don't just trust an app or a book. Get multiple sources of confirmation. There's even great Facebook forums you can use.
Excellent. Okay. Well, that is a ton of wonderful information for us to think about, and we are going to thank our wonderful scientists, and we're going to move on to another section of the evening, but let's give a round of applause for our scientists. Thank you. Grace, Damon, and Gordon. Bye. Goodbye. We will miss you. You can find them in the lobby later for all your questions. All right. Next up, I would love to introduce you to Gabriela Quirós. She is Deep Look's award-winning coordinating producer. Gabby, can you come on and join us? Check out this outfit, everybody. So we are lucky to have Gabby here with us. She's going to talk to us about how we actually make videos like the ones that we just watched. Welcome, Gabby.
Hi, Laura.
Nice to see you again.
Nice to see you.
So can you start us off and tell us what some of the challenges are in filming fungi for Deep Look?
Well, the big challenge is that they don't really do anything. So for people who know our show, we focus a lot on animals, and we focus on their behaviors. And what we really want to do is like show people an interesting behaviors that an animal is doing to make a living. And so with fungi, that isn't as obvious. And so that's why in the videos that you saw, we chose fungi that were doing something or that we could show being active. So for example, the bird's nest fungus shooting out the spores when the raindrop falls on them,
yes,
or
adding all those fun words like zap pow.
Exactly.
Things like that.
The cage fungus, using those time lapses, you can see it growing from that egg into the cage.
Yes, I mean, one thing that we often talk about in the Deep Look meetings when people are pitching is like, does it have eyes? Does it have a face? And mushrooms do not have those things. So, yeah. So we try to find action, like what Gabby said. So how did you film the spore sacs that were flying out of the bird's nest? Fungi.
Yeah, so I see we have here up on screen a photo of our room upstairs where we were able to do filming, and I brought a prop, which is a burette. And so for that particular episode, what we did was try to kind of do what the researchers had done in order to film the spore sacs. And so I spoke to Nicholas Money, who had done this research with his colleagues, and basically he said, get a burette and fill it up with water. And in the paper it said that they had set it 1.2 meters above the mushrooms, and that they had the most success, or that the spore sacs had been ejected most often when the drops fell right on the rim of the cup, so not in the middle of the cup, but we should try to aim for the rim.
Super easy to do,
yeah,
right?
Exactly.
It's really simple.
And so you can see the burette there at the top near the fluorescent, and then there's an apple box, which is that wooden box, and I'm in the back there, but when Josh Cassidy, our series cinematographer, focused in on the mushrooms, then I stood on the apple box, and it was my job to turn this and make the little drops fall, and he would guide me, a little bit more this way or that. And we basically spent a day filming in this room.
And you up on those boxes just going?
Yes, and just trial and error. And Eleanor, who will be up on stage later, who's in green there, she brought boxfuls of these mushrooms from her backyard, which is featured in the story. We also then went to her backyard and filmed, but the first day we were here doing this basically lab work and creating these little sets, which you can't see very well there, but we tried to create a little set that mimics kind of the mulch bed, basically. And then when we went to Eleanor's house, then we brought a high-speed camera, which we didn't have here, but it's that camera is a Phantom Veo, and it's a high-speed camera, and it needs a lot of light, and so in this, I wanted to show you that we have 1, 2, 3, 4 lights, and it's daytime, and it still was just barely enough light to be able to film these in high speed.
Cool. It's awesome to see the behind-the-scenes footage. Yes.
Yeah.
Thank you. And so how did Josh film the time lapses for the cage fungus episode?
Right. So that was a fun challenge. And here's Damon, who you all saw just a minute ago. And that, if you can see at his feet a little bit in front of him, is a very small kind of golf ball cage fungus, a young one, and he's extending his arms to show you how much mycelium is underneath that is producing that single egg, as it were, and this is a little bit of that mycelium. And then basically what Josh did with Damon's help was kind of excavate these eggs with some of the mycelium and then bring them back here to KQED and set up the camera or several cameras and with pointing to several of these young cage fungi, and then have them taking an image every so often and see, again, like trial and error, see which ones had produced the best sequence of images. And that's how he was able to film the time lapses. Some of those time lapses were filmed by other filmmakers, but at least a couple of those were ones that Josh was able to film. It's very laborious to film time lapses because there's a lot of trial and error that happens, and so that's what makes those episodes so special. And I think that's why people respond really well to time lapses. People love the passage of time, especially if it's like something so amazing as going from a very small white egg to this crazy red thing that unfolds.
Yeah, yeah. And I think also what's really powerful about film and a component of deep look and other videos is just the use of music in that one. I hear that sort of like orchestral or that choral kind of music with the time lapse is really beautiful. So how did you come up, when you want to do an episode about fungi, how do you come up with the ideas for these episodes?
So we often rely on naturalists like Damon, for example, to suggest ideas. We follow our own curiosity or our kids' questions. I mean, everything is fair game. It can be, we often return to scientists who we worked with in the past and see what they're working on. People approach us, or scientists will approach us with their newer research. And we're very fortunate to be able to work with scientists because it is very time-consuming what we do. And so I wanted to show you a few behind-the-scenes photos. So this is Carolyn Elia, and she was a scientist at Harvard who we worked with to film the flies that were infected by the fungus that you saw at the very, very beginning. And she has an incredible setup in her lab where she has these flies, and that have been infected with the fungus, and she has them on a system where she knows exactly which flies are going to start expelling the spores when. So it was very convenient for us to be able to film with her, because she could provide us with flies that were going to do X, Y, or Z in exactly an hour. And so it's like your dream situation.
Yeah.
And then here's, I just wanted to show you for scale. This is the top of a toothpick, and there's a fruit fly at the very top. And it's inside a tiny glass box so that the fly won't fly away. And here is Anne Pringle, who we worked with to film the death caps. And one of the things that we're able to offer scientists in exchange for the days that we spend in their labs filming is oftentimes they haven't had the opportunity to see the animal that they study very, very close up. And it isn't infrequent that a scientist or a naturalist will tell us, I never knew that it looked like this up close, or I had never had a chance to see it. So that's kind of a small gift that we can give them in return for their efforts.
Awesome. Well, thank you, Gabby. And let's all give Gabby a round of applause.
Thank you.
Don't forget your prop. Congratulations. She has won that for participation tonight. Okay, so next up we are going to bring up Reggie Harris, Tomas Garrett, and Emily Savage to the stage. Reggie Harris is the founder of Oakland Hyphae and Hyphae Labs. Tomas Garrett is the lead chemist. Emily Savage is the analytical chemist. So welcome, everybody. Thank you all for being here. Okay, so Oakland Hyphae is a leader in psilocybin mushroom potency testing and research. Can you just tell us what that means?
Yeah, so thank you all for being here. It's an honor for us to be here. So just to answer your question, I came to this space through selling mushrooms, and so we would have conversations. Tomas was there with me in the beginning. We'd have conversations, and we would be growing these mushrooms for folks over in Oakland, and we'd be trying to sell the mushrooms, but there was no quantitative data that we could put to valuing, how strong the mushrooms were. Some people wanted to microdose, but mainly people just wanted to get high. And so there was anecdotes, is it blue? Do we want just caps? Do we want just stems? But this makes doing business very difficult, trying to hit a moving target.
Yeah.
And so myself, Tomas, Emily went to work trying to quantify potency, because that's at that point what people put value on. And they developed a method. We had the method second party validated through other labs, and the rest is history. So we were the first people to do this, and now there's a bunch of labs out here doing it, but it just helped us, quantify what we define as potency, and now we're actually having conversations about actually what is potency. We've expanded our panel of tryptamines from just psilocybin and psilocin to 13 other tryptamines, and so we're talking about like entourage or ensemble effects, how these tryptamines might work in tandem to produce what we call, a trip.
Yeah, that is super interesting. Another aspect of one thing that you all do is also the psilocybin cup and the Oakland Psychedelic conference. You curate those things. Can you tell us about those events?
Yeah, I'll talk about these and then take a step back to let the scientists. We got a science-heavy crowd here, so I brought the scientists. I'm the business person. But so once we developed the method to test mushrooms for potency, we had to figure out. I'm a business person, and we had to figure out how we would monetize what we've developed. So I have a background in community organizing, which you can look at it as activism, or you can look at it as politics, whatever. You organize people. So the first thing I thought was let's organize an event around the testing. So the psilocybin cup was inspired by something that they did in cannabis called the Cannabis Cup, or the Emerald Cup, that was developed up there in Medicinal County. And what we did was 4 years ago, you remember, we actually had to buy the first submissions to, like, we would go around to sessions. In Oakland, there's something called sessions where they're underground markets where you can get like weed or mushrooms, and we would go to these sessions and buy samples for the first psilocybin cup. So we were paying people for the pleasure of for participating in something that what we found would like create careers for people. And so we ran the first psilocybin cup a few years ago. It was a hit. What we figured out was that we were the only people in the country who were who had access to this many cultivators, and we would put a list together. And then, you know, as this psychedelic renaissance or psychedelic shroom boom, I hate that term, but as it would develop, you'd have people who were looking to cultivate or looking for genetics, and you know, our source was the only place that people could go to say, okay, well, I know that there's strong genetics here, or I know that they're growing this genetic over here. And so we've been doing this now for 4 years. There's been many people that come behind us and try to duplicate what we do, but as we said this year, our cup is the only cup that matters. So.
Okay, great.
And just really quickly, the conference with the Oakland Psychedelic Conference, we started bringing people together culturally. Tomas and I met at a decriminalized nature meeting in 2019, and we would be going to these meetings all the time, and I wouldn't see any brown people at all in these. It wasn't culturally pleasing, but I would go because I was very interested in the mushrooms. And so once I kind of got my legs up underneath me with the psilocybin cup, it was my mission to create a space that did feel comfortable for me to hang out in and talk mushroom shit, and for people that look like me, you know, because I don't have letters behind my name. I'm not a biologist, I'm not a chemist, but I am a mycologist, you know, a self-taught citizen scientist. And so this is what we've created. We've had the Oakland Psychedelic Conference. We've had the California Psychedelic Conference down in LA. We've had one in DC. And coming up this year, we're having the International Psychedelic Conference in Negril, Jamaica. So.
I do want to hear from all of you if you could tell us your mushroom origin stories.
My relationship with mushrooms is so intertwined. Reggie and I were in the lab, but I met him at a decrim nature meeting, and I really fell in love with mushrooms when I was working at this cannabis lab because my background is in chemistry. I was doing pesticide and mycotoxin testing, actually, for cannabis and residual solvent testing, and I found out about this decriminalized nature meeting. We go there, we found some people who grew the mushrooms, we found some people who were investors who wanted to find out what's going in the mushrooms, and then me and Reggie go to Jamaica at the first iteration of Hyphae Labs, and then now here we are 4 years later on stage talking to you about mushrooms. And so this is my whole life. I dress up like a mushroom and I talk to everybody about mushrooms. Life is really good.
Yeah, so I grew up in Western Pennsylvania on a cannabis farm in the 90s. So I was around mushrooms from a pretty young age. But I moved to California in 2014 and started working in cannabis testing, mostly microbiological testing, for about 5 years. And about 3 or 4 years in, we had to start testing and identifying fungi that were growing on cannabis. And I ended up spending hours under a microscope looking at plates of fungi, and that was kind of it for me.
What about you, Reggie?
Yeah, so for me, it's not science, it's commerce. So Well, no, it was curiosity. So in high school, Eminem had this song about mushrooms. I was just obsessed with hallucinogens at that time, and I was consuming a lot of trying a lot from like nutmeg. And if you've ever taken nutmeg in high doses, it is a hallucinogen. But I found, I came across a really great source for mushrooms when I was able to find them. And at that point, like none of my black friends was doing mushrooms at all. So I ate them with my white friends. I'll never forget, it was raining, kind of like it was today. We ate them under this deck. And people say that mushrooms communicate with them. I say mushrooms communicate with people in different ways. But the download that I got in high school, the guy said, "Bro, how much for an eighth?" And I didn't know. I did the math. I figured out I could make a profit. And from then on, the relationship with mushrooms for me led me around the world. I went to college in Florida. I dropped out of college halfway through Florida to go work at the largest mushroom farm in the world at that time. And then fast forward to 2019, like we said, we linked up and the rest is history.
So how are mushrooms used for psychedelics?
How are mushrooms used for psychedelics?
Yeah.
It's different for everyone. Some people prefer to microdose them and have them only in small quantities, and some people swear by only eating at least 10 grams. And what their intention is often very personal and spiritual, and sometimes it's related to revisiting a personal event or traumatic event in their life or ancestral trauma. Sometimes it's trying to reconnect to their joy and their happiness. And research is telling us a lot of things. There's a lot of medical research to suggest things related to anxiety and depression and other mental health outcomes. Our research related to psilocybin testing specifically is telling us that there's a lot that we still don't know, and that there's a huge range in potency that we're seeing with mushrooms, and it's often not very predictable. And that's why it's good for people to be informed about how much they're ingesting.
For me, it got me off of Adderall. So and a lot of people are talking about this. I stumbled upon this as a solve. In order for me to maintain a regular job in the regular workforce where I had a boss, I would have to take Adderall every single day, and Adderall would ravage my body. And I stumbled upon, just experimenting with microdoses and making stacks with lion's mane and a little bit of magic. I learned that that I was able. I haven't touched Adderall in years at this point, and I'm able to manage multiple businesses. But so for me personally, it was a solve for being more or less hooked. I wasn't abusing it. I needed it to do my work. But now, I can mix magic and lion's mane. And we got the clown here.
I'm wearing a mushroom hat.
Go ahead. How do you?
He's wearing a mushroom hat with a mushroom in it.
I break it down into 3 levels. There's many applications for using mushrooms, but I like to think of it like a tree. At the top, you got where the leaves start to dance, and this is generally around 1 gram or so. At this point, colors are brighter, sounds are crisper, and it's a low dose. That's what people are using it for at that level. At level 2, the trees start to breathe and the ground starts to move. At this point, maybe you don't go to an event like this. Maybe you need to be at home. Level 1, here's fine. Level 2, hang out with your dog or something. And then level 3 is when you're tapping roots. As Emily was saying, you can go back to really traumatic or potent experiences within your past. And when you take large doses of mushrooms and you reach level 3, you're not getting higher. You're actually going more in. You're going towards your roots. You can actually tap in with your ancestors. You can tap in with things that you haven't addressed in a while. And if you really go in, you can hop out of the matrix. But that's a whole nother story. That's those 10 grammars. That's, go, just look into it. Back of his shirt.
Just to wrap up this section, talking about psilocybin, can you tell us sort of where are we in California with legalization, and even on a smaller level in Oakland and San Francisco. Yeah.
So
what's the status?
Right now in Oakland and in San Francisco, both of these municipalities are decriminalized, which means that it means that you can still go to jail, but it's lowest priority. So if somebody's jaywalking, the person right now that's jaywalking gets reprimanded before me carrying my mushrooms across the street.
And that's to have, like, to possess, and also not to sell them.
Yeah, sales is prohibited at every single level. We saw last year SB 58, which was proposed by Scott Wiener. It got shot down by Gavin Newsom. Newsom says that he'd like to see some framework put in around, and I'm summarizing, but around like consumption for harm reduction. So, you know, for facilitators, he wants to see just framework. He doesn't just want to see a free-for-all. So I was actually on a Zoom call today with a bunch of activists that are going to be launching whatever the new iteration of SB 58 is.
Okay, for this legislation.
Yeah, and so it would decriminalize mushrooms statewide. It wouldn't create a path for commerce, but it would decriminalize mushrooms. It would allow for ceremony holders to be able to do that sort of work without fear of being prosecuted by the government. There's one initiative that's kind of sunsetting right now, and it's the psilocybin mushroom initiative that's being put forth by Decrim California. It doesn't look like they're going to get the signatures that they need. And then the one last initiative that will be on the ballot is something that I'm actually involved with. It's called the Psychedelic Wellness and Health Initiative, which would decriminalize a number of psychedelics, not just mushrooms, but we're talking about MDMA, ibogaine, and we could go on. We could go on and on. DMT, but it would decriminalize a lot of these things and then create a framework to facilitate sales.
Okay, great. Well, thank you all so much for being here. Yeah, thank you. Let's give them a round of applause. All right. Thank you, guys. Okay. So now we're going to move on to the portion where we talk about eating mushrooms, not psilocybin mushrooms right now. So I'd like to invite up Chef Chad Hyatt. Yeah, let's hear for Chad. This is Chad. Chad, if you could go to your chef's station over there, please. Thank you. Chad is the author of The Mushroom Hunter's Kitchen, which you can buy at his table after the show. He's both an expert forager and a classically trained chef. He's cooked in a variety of restaurants and private clubs around the San Francisco Bay. He teaches classes. He does all sorts of things. We're also bringing on Gordon Walker, bringing him back, our scientist, to cover the science, as Chad shows us, his mushroom ceviche. We are also going to be passing this out to you to eat this while we discuss how to actually make it. All right.
So I'm starting into something here because I have limited time, so I'm gonna work while we talk.
Excellent. That is what we're hoping will happen.
So first, Laura told us that I'm gonna do a ceviche for you, and I want to point out that typically ceviche is done with fish or seafood, and what happens is you put and I promise I'll explain what I'm doing here in a second. But typically you take some sort of fish or seafood and you put lime juice or some sort of other acidic liquid on it, and that kind of cooks the fish as it marinates. That doesn't work with mushrooms. With mushrooms, they get their structure from something called chitin, which is what gives structure to insect shells and the like. And lime juice isn't going to break that down. So when I'm making ceviche with the mushrooms, the first thing I did, which I'm not showing you on stage here, but the first thing I did is I actually boiled these mushrooms. Mm-hmm. And
they were these. This is the beach mushroom, or Hypsizygus.
Yeah, and it's actually a mix of the brown and the white, which are the same species, but they're just different cultivars, like different kinds of tomatoes. Mm-hmm. So all I have here, I just have the mushrooms that I boiled, mixed with some lime juice, and I just cut up a little bit of a serrano pepper. I'm not going to make this too hot because I hope you'll taste it in a moment. I don't want to hurt you that badly.
Oh, thank you.
But anyway, I like to show people this because people are always taught that you shouldn't treat mushrooms, you shouldn't get mushrooms wet, you shouldn't wash mushrooms in water, you shouldn't cook mushrooms with water, and I think a lot of this comes from kind of old wives' tales, just misunderstandings of what
Bad advice from Julia Child. Don't listen to Julia Child about mushrooms. She was right about everything else, but not mushrooms.
Oh, and I almost forgot. It needs a little bit of salt, so I'm going to just put a little pinch of salt in there, and we'll let these marinate while I talk. But again, with mushrooms, just like mushrooms are not fish, they're also not plants. We're used to vegetables, where if you boil a vegetable, it gets its structure from cellulose. You throw it in the boiling water. Cellulose is water soluble. The vegetable, if you forget about it, will disintegrate and you will never find it again. Mushrooms, once you cook them through, the cell walls burst, that's the end. They won't keep cooking. They don't get any softer. They won't disintegrate. They'll give up more of their flavor to the water after a while, but you're not really worried about losing them to the seas.
I have a theory about mushrooms, and I'm curious to ask you about this. So I think they're almost like, texturally, they're almost the opposite of meat. Because when mushrooms are raw, they're really brittle, and you can snap and break them. Whereas when meat's raw, it's like, ground meat, right? It's tacky and sticky. And when they're cooked, it's almost the opposite. That cooked meat can like shred, and you can pull it apart. Whereas cooked mushrooms are now kind of like tacky and sticky and can be like blended. So do you feel it like that makes this perfect for the ceviche dish kind of thing, right?
That is, I don't know how to respond to that one. It's an interesting observation for sure. I mean, certainly in regards to cooking something like this, a lot of the things you might do with raw meat, I would do exactly the opposite, where I would do them with a cooked mushroom because texturally, it's not just about breaking it down to make it digestible or something. It's just texturally, it's going to be more pleasant if you cook it.
And are there mushrooms you can eat raw?
So there are a few mushrooms you can eat raw. Everybody's probably seen too many white button mushrooms on the salad bar.
Yeah.
It's really one of my least favorite ways to eat any mushroom.
But pretty flavorless.
Yeah, not very flavorful, and there are a lot of people these days who will argue that it's not very healthy either. That there are some chemicals in there that are not so good for you. I don't know how true that is.
Don't worry about the chemicals in the button mushrooms. In like a wild agaricus species, you might have something problematic, but in portobellos and white button mushrooms, they're safe to eat raw. I just don't like eating them.
Yeah.
I just don't think they taste good. They're better when you cook them.
Yeah, so what I was getting to is most mushrooms, wild mushrooms, most, like these beach mushrooms that I'm doing here, you don't want to eat them raw. They're largely indigestible. The chitin that I was saying won't break down from lime juice also won't really break down in your digestive tract. That's why you want to cook it first. There are a few mushrooms like porcini that are traditionally sliced thin and eaten raw, but even those in small quantities, people don't typically eat them like an apple.
Yeah.
And even then, they usually marinate them some olive oil and some salt. So you kind of break them down a little bit with just how you're preparing them, right?
Yeah.
And since it's hard to really quantify what a mushroom is, right? It's not a vegetable. It's not a fruit. It's
a mushroom. They're their own thing.
It's a fungus. It literally is its own thing.
So, what kind of nutritional value are we getting from them?
So the first part, I'll defer a little bit to Gordon on this, but first, mushrooms have lots of dietary fiber, which is really nice. They have, depending on the mushroom, they have different kinds of vitamins and minerals and such. There is some amount of protein. I don't know how much, and I think it varies quite a lot. Again, mushrooms are a whole kingdom. It's, like saying, what do you get out of a vegetable? You have to be more specific. So it's... Again, I'll defer to Gordon to get more specific.
There's a lot of, as you said, there's nutrients, the micronutrients of vitamins. There's a good amount of high quality protein. It's nowhere near as much as meat. It's easily 1/10 the amount of protein as a piece of meat, but you get really good protein. So because fungi are more closely related to animals than they are to plants, the nutritional profile of the amino acids in mushroom protein is actually better for you than eating a plant. You eat plants, you have too much lysine. Whereas you eat these, it's pretty much a 1-to-1 mix of amino acids is what you need for an animal. And like Chad said, you get a lot of really good dietary fiber, which is something that we think about how important dietary fiber is for us, because it's stuff that not only helps us be regular, but also helps stimulate a good gut microbiome, which also leads to better like cognitive and immune function. So eat more mushrooms.
And another one, in terms of vitamins, a lot of people have talked recently about vitamin D, where mushrooms, like animals, if you leave them in the sun, you can actually leave your mushrooms in the window for a few hours and they will produce vitamin D just like you will.
Same biochemical pathway.
So eat them and get your vitamin D.
Yeah.
Wow.
Basically.
That's awesome. Okay, so you are quite obsessed with mushrooms and cooking mushrooms, right?
Yeah. So
can you tell us what is your origin story?
Okay, so my origin story, not so surprisingly, started with cooking mushrooms. I went to a fancy dinner that at some restaurant many years ago, and I started talking, the supplier of the mushrooms was there, and I started talking to him, and he convinced me that all I needed to do to go pick my own mushrooms was buy a book and go out and start picking mushrooms. And being kind of naive as I can be, I went out and bought a book and started picking mushrooms. And I really, what kicked it off for me was there was one particular hunt I had up in the Lake Tahoe area where I had gotten some intel from somebody that porcini were popping around Tahoe.
Nice.
And I skipped out on work the next day and I convinced a friend to do the same, and we went up there and there were porcini everywhere. And after that I was totally hooked.
Wow.
What did you cook?
Oh God, I don't, that's another long conversation.
It's like the Easter egg hunt theme though, again.
No,
back.
absolutely.
It's so exciting.
I think Damon, brought up before the Easter egg thing, and I was always an outdoors person, and learning about mushrooms just made it so every time I'm outside now, it's a scavenger hunt.
Yeah.
So.
That's awesome, thank you, yeah. Another question that I've had is actually where our mushrooms come from. That, like, if we buy them in the supermarket, if we get them at a farmer's market,
So most of the mushrooms you get in the store, like these guys are fairly exotic for this store, but they're pretty typical in kind of Asian markets, and a lot of people are cultivating these. Most mushrooms you get in the store are cultivated. The button mushrooms, portobello, cremini are all actually the same species, believe it or not.
Oh.
The shiitakes are cultivated, oysters are cultivated. Most of the things you see at the store are cultivated. And there's, I saw you mentioned a little bit before about kind of mycorrhizal mushrooms in one of the videos. Mushrooms kind of occupy a few different ecological niches. And mushrooms that grow by kind of eating decaying matter are easy to cultivate, relatively speaking. Some easier than others. And so all the cultivated mushrooms are that group. Like the shiitakes and oysters grow eating dead wood, so people make compressed sawdust things and spawn them out of that.
So that's what's called saprotrophic. You heard that in the first video. And yeah, pretty much all that, as Chad said, is like grain and sawdust, except for all the button mushrooms and the portobellos. Those are usually grown on sort of fermented animal dung.
And so, so if you're using button mushrooms, portobello and stuff, they're sterile. They're grown on sterilized compost, but that's a fancy word for animal dung. So wash those.
Okay, so
anyway,
in contact with water.
Whereas the rest of these actually come in quite clean. You really don't need to wash most of the ones you're going to buy at the store unless it's a portobello. Whereas the black trumpet that Chad's handling is literally full of dirt all the way down to the base.
Right. But so back to the question, to bring it back to the question. There are some mushrooms that you will get that are only wild. Mycorrhizal mushrooms cannot be cultivated because they have symbiotic tree partners. Without those trees, you can't grow them.
And that.
So this is an example like porcini and chanterelles and truffles. Although truffles, they are actually inoculating orchards now. But these black trumpets, I brought the black trumpets because I am going to give you guys another little taster when we leave with something from the black trumpets. But these mushrooms are only come from the wild, so they're not cultivated. But 99% of what you see in the store is cultivated.
Wow, that is...
This is the time of year, though, to go to a fancy market, though, and see chanterelles, see black trumpets. The only really ectomycorrhizal mushroom that they've been able to cultivate with any success is truffles, and it's the only one that makes enough money for it to be worth it for them to do that. But even then, the cost of truffles is pretty high.
All right. Any last notes that you'd like to tell us about this ceviche? What else are you going to put in?
Okay, so let me show you how to finish this ceviche. So remember, all I have in here is just the boiled mushrooms marinating in lime juice with salt and a little bit of chili. I have, so I'm gonna move this plate over here, but I have some thinly sliced red onion. And I have some cilantro on my board that you just saw me chopping. To finish, I'm just going to add these to what I already had marinated, and try and keep my hands from stinking like onions more than they already do.
Chet, do these mushrooms bring a lot of their own flavor, or is you really just depending on how you season the ceviche for the flavor?
So let me come back to that in a second. So so again, all I'm doing is I'm mixing these in. I think it's an important point that this method for ceviche is traditionally from Peru. I learned this from Peruvian guys. Peruvians are famous for their ceviche. I learned this from fish. The only difference in what I'm doing is I'm doing it with boiled mushrooms instead of raw fish. It's important you marinate the stuff first, and then you're putting the onions and cilantro in at the end. Okay. The samples you guys are getting, they've been soaking for a while because it's the only way we can really serve it to you here. But you'll get much more, the flavor pops a little more, you get more crunch out of the onion, and the flavors are all kind of separate. The last thing, let me move this so you guys can see what I'm doing here. The last thing is, with the Peruvian ceviche, they'll always serve with a little bit of boiled sweet potato, and the starch kind of cuts through the acid of the lime juice, and it also helps with the heat because they tend to like it pretty spicy. So I just want to taste this and make sure the salt's okay.
Remember to taste your food.
Yeah, if you don't taste your food, if it tastes good, you got lucky. And in this case, I got lucky because it actually tastes good. I'm not going to do anything to change it. But all I'm going to do, again, I just mix this stuff up. I'm just going to serve it with the boiled sweet potato. The Peruvians always keep all that extra juice in there. They call leche de tigre, which means tiger's milk. And that's part of the kind of experience of eating the ceviche. Like when you're with Peruvians, somebody will usually have the job of drinking the leche that's left in the plate.
I love doing that, actually.
Yeah.
So, it is I enjoy that.
So I always have to make you taste it.
Okay,
I know
awesome.
I like it, so.
All right, well, what did you all think? Yes? Applauso.
So I will tell you, while you're tasting that, I do have one more taster for you guys that I'm not going to talk too much in detail about.
It was so good.
But I mentioned the black trumpets, and I like to show people things beyond just sauteing mushrooms and tossing them with pasta. I want to kind of bring you out of that and show you can do other things. I have another taster for you that's a jam, actually, made with the black trumpets.
Wow, not something I would expect with mushrooms.
Exactly.
Yeah.
And
cool.
the jam is really simple, just the mushrooms cooked down with a little bit of sugar and a little bit of vinegar, and that's it.
Nice.
So...
And so can we find these recipes in your book?
Cookbook full of
recipes
recipes that are lots of unusual uses of mushrooms.
Awesome.
But hopefully all tasty. I try not to do things just for shock value.
Okay, well, thank you. Let's all hear it for Chad. Thank you so much. Thank you. We'll see you outside. You can find him and his books outside. Okay, our final guest for this evening is Eleanor Raab. We'll bring her up. She is a naturalist and a TikTok creator who posts videos about foraging where you live, and she can help you find cool mushrooms in your own backyard. This is Eleanor. We wanted to conclude with Eleanor because now that we've seen all these wonderful things, we want to give you some tools to go out and do your own foraging, your own Easter egg finding. So thank you and welcome.
Yeah, thank you for having me. I'm excited to be here.
Yeah, okay. Well, so briefly, can you tell us about your mushroom origin story?
Yeah, I mean, funnily enough, when I was a kid, I actually really hated mushrooms. I did not think that they tasted good at all.
Were you eating the the white button ones, or?
Yeah, exactly. But my mushroom origin story actually starts with one of the mushrooms that we saw in our Deep Look videos. I found some bird's nest fungi in my backyard, and I was like, what on earth are these things? And I don't know, I was maybe 10, 11 at the time, and I've always had an interest in the natural world, but it kind of just spiraled from there, and I've just been learning more and more about mushrooms as I've grown older.
Cool, thank you. And actually, I think you were the person who came and pitched that story to us, right? That you're the reason why that video exists.
Yeah, I had such a passion for the bird's nest fungi, and I found Gabby, pitched the story, and you might have seen me in some of those pictures, and you also would have seen my backyard in the bird's nest fungus video.
Yeah, thank you for letting us film in your backyard.
Of course.
Okay, so we have watched a film about death cap mushrooms. How do we get started foraging without killing ourselves?
Right. So to start with foraging, I would say the first step really is to go out and learn about mushrooms honestly, without thinking about what you're going to eat or not eat. I think it might have been Gordon who said something similar, but once you get out there and you start learning how to identify mushrooms, any mushrooms, it'll quickly become pretty clear, which ones are easy to identify to eat and, which ones are deadly. But, there's a couple of safer mushrooms to start with. I would say that chanterelles are a really, really easy edible mushroom that grows pretty prolifically in our area that is kind of hard to mess up with, there's not a lot of deadly lookalikes.
Yeah, they're visually easy to identify.
There's one lookalike that will maybe make you sick to your stomach, but it won't kill you.
Yeah, okay, great, yeah. And so tell us about etiquette around mushroom foraging.
Yeah, for sure. When you're foraging, there's a couple of things to keep in mind. Primarily, I think the most important thing is to be a really good steward of the habitats that you're foraging in. And this goes for all types of foraging, not just mushrooms. And so I think that being a good steward involves, taking only what you need. Because you can go out there and get really, really excited, collect 10 pounds of mushrooms, and then you get home and half of them are going to rot on your counter. So you really only want to take what you're going to eat. You also want to be mindful of the habitats that you're in. You don't want to trample over sensitive plants while you're, trying to get to the mushrooms that you want. And you also want to, take only a small section of any given mushroom grouping. So a good rule of thumb is to take about a third of the mushrooms and to leave the really old ones that are too gross to eat, and to leave the really young ones that haven't had a chance to disperse their spores yet.
Oh, that's really helpful. Okay, and then around that, one thing that has come up, I've heard people talk about, is like feeling bad about actually picking a mushroom because it almost feels like you're uprooting a plant. But actually what we've heard tonight is that this is really the fruiting body of something. Mm-hmm. And so you're not hurting the mycelium beneath it, right? If you pick that mushroom?
Yeah, for sure. Yeah, you aren't, if you're really aggressive with it and you dig up all around the mushroom, sure, you're gonna hurt the mycelium, but no one's doing
your
that.
foraging.
Yeah.
Yeah, okay,
yeah.
Cool. And where can we find mushrooms?
Literally everywhere. One of my favorite hobbies is going on walks in my neighborhood and finding as many mushrooms as I can. And I went on a walk the other night and I think I identified at least 30 different species. And it was maybe a 1.5 mile walk around suburban Menlo Park. So they're in your backyard, they're on the streets, they're in your local parks. If you want to find some of the more exotic looking ones, you may have to journey to some of your local open space preserves or state parks, but the Bay Area is a great place for that. We have, I think, over 1 million acres of protected land in the Bay Area now, so yeah, lots of places you can find mushrooms.
Yeah. And what's awesome is just really, I think you've all probably checked out the tables as you came in, but be sure to look at them again as you go out, and some of the things on there I would not have noticed. Some are just so small, like this one that you brought in, right? This.
Yeah.
What do you call that one? Can you talk about that just a little bit? Like.
Yeah, I brought in a really tiny mushroom called. You're talking about the eyelash cups, or they're called eyelash cups, and they're I don't know, millimeters. They're tiny, but I found that one in my neighbor's front yard. So they're everywhere.
That's really awesome. So Eleanor has done something really wonderful for us. She's created a whole list of resources which were emailed to you before this and will be re-emailed to you after this. Can you just walk us through some of those things so people know what they now have at their fingertips?
Yeah, for sure. So in this little guide that I created for you, it starts with vital tips. So basically things that you should remember while you're out there looking for mushrooms. One, obviously, consult an expert before you consume any wild mushrooms. 2, harvest sustainably, so that's that rule of thumb that I was talking about. 3, respect private property, and then also follow the regulations of your state and local parks. Foraging is pretty heavily regulated in the Bay Area, so you really need to know what parks you can and can't forage in. And there's actually only a couple of parks that you are legally allowed to forage in. And I've put a list of those for you guys in there as well. So that's going to be Point Reyes National Seashore, Salt Point State Park, SoCal Demonstration Forest, and Jackson State Demonstration Forest. And some of those require permits, and some of those have bag limits, so you're only allowed to take a certain amount out in any given day per person. And then I've also put a list in there of where you can find other local and state and county parks that you can go looking for mushrooms in, but not forage in.
Okay, great.
Yeah.
Well, one thing that I think is so awesome about the knowledge that you bring, and that everything that we've heard this evening, is to go out into the world and to look at it in a slightly different way, in a closer look, which is also a goal that we have with our Deep Look video series, to see things just a little bit differently.
Yeah, I mean, I would say the best way to get started if you want to learn how to identify the mushrooms that grow in your backyard is to hop onto iNaturalist, which I think other folks have mentioned tonight. It's a free application, completely free. It'll never charge you anything. Hop onto iNaturalist, and you can actually, look at the mushrooms that have been identified literally in your city, maybe even on your street. And you can go walk and go to that site. Maybe the mushroom will be there. But it's kind of like a list of what you can find in your neighborhood. It's a really amazing resource. And you can add to it too. So you can take pictures of the mushrooms. You can upload them. It'll help you identify them, or at least get you closer to the genus. And then there's a lot of community science projects that happen through iNaturalist. So you can be a community scientist just by going on a walk, looking for a mushroom, snapping a picture.
Well, thank you so much, Eleanor.
Yeah, thank you for having me.
Yeah, let's hear from Eleanor. You in the front. All right. I want to reiterate that all these really incredible minds that we had on this stage, you can find them outside afterwards, so you can ask them all the other questions that you might have. And that wraps up our stage portion of the evening. Many thanks to all of our truly fantastic speakers. And if you love the Deep Look videos that we screened, you can watch more of them at kqed.org/deeplook. Also on our YouTube channel, it's KQED Deep Look, and you can subscribe so you can know when new videos come out every few weeks. If you really love the show, please consider supporting us on Patreon, and you get exclusive behind-the-scenes merch and updates and all sorts of cool things like that. And now I'd like to invite you all to just mingle with each other and mingle with the scientists and the experts that we've heard on stage here tonight out in the lobby. And thank you so much for coming and making this such an awesome space this evening. Take care. See you out in the lobby. Bye.